All right, cool. All right, so this is Unit 7, topics 4, 5, and 12. All right, this is Hardy, Weinberg, equilibrium, and population genetics. This is where we're going to get some math in. So, what is a population? So, a population is a group of individuals of the same species that live in the same area and interbreed to produce fertile offspring. Okay, so a population, everything's the same species. You can mate, you can make babies. It's great. The gene pool is what we refer to as the whole population's genetic makeup. We're going to get to that on the next slide. But when we talk about increasing the diversity of the gene pool, this is what we mean. It's the population's genetic makeup. Consists of all copies of every type of allele. If there's only one allele present for a particular locus, then the population is fixed, right? So if there's only one allele that codes for fur color, right, then all of the individuals in that population will have the same fur color. We call that a fixed population. If we have many fixed alleles, that decreases the genetic diversity of our species and of our population as a whole, and eventually leads to bad things for the population. So the opposite of this, so if you have many alleles, then you have greater genetic diversity and your population's much stronger. Pause here if you have to take notes. Cool. So let's take a look at the gene pool of this mouse population. So we have a population of mice. We have a little bit of genetic diversity here, but all of these mice are from the same population, meaning they can mate with one another and produce viable offspring. So, so this guy can mate with this gal, and everything will be fine. They'll have viable offspring. So when we talk about gene pool, right, so you can see the black mouse here is homozygote dominant, heterozygote, homozygote recessive, and then homozygote dominant once again. We can see that their gene pool looks like this. So because of independent assortment of alleles, we separate these. So we have big B's and little b's, but this is what we call the overall gene pool. It's taking all of the alleles, whether they are dominant or recessive, and tossing them into this kind of mishmash of genes. Boom, flipping it all around. So let's talk about population genetics. So the frequency of alleles is going to change over time, right? How often a certain trait shows up in an individual is going to change over time. Remember that populations evolve, individuals do not. Again, what we mean by that is you can't take an individual mouse, right, and change its, at least in natural selection, you can't change its individual. You can't have it evolve into something else, but populations as a whole evolve. So it's that passing on of heritable material. We can talk about microevolution, that's small scale genetic changes inside of a population. Evolution's driven by random occurrences, most of the time random mutations, but also things like outside environmental pressures as well. Mutations, here we go, genetic drift, we're going to talk about genetic drift in a second. Migration and gene flow, so some species of animals will migrate, and of course natural selection. Pause here to take notes. So let's talk about each one of these individually. So mutations, we've talked about this in Unit 6. A mutation can result in a genetic variation. So if we change the genotype, we change the phenotype. This can form new alleles, and natural selection can act on the varied phenotypes. Mutation rates tend to be slow in plants and animals and faster in prokaryotes due to a faster generation time. That makes sense. It takes plants some time to grow. It takes animals some time to make and have babies, but prokaryotes can just asexually replicate on their own. So it tends to go faster in prokaryotes than in multicellular organisms. Remember, though, that mutations can be harmful, neutral, or beneficial, right? Most mutations are neutral or the harmful range, but not all mutations lead to evolution. We know this. If we have a nonsense mutation in our DNA, then it's going to produce a stop codon. That protein's not going to work, so that's a harmful mutation. A silent mutation would be neutral, and then some mutations become beneficial for the species as a whole. We'll look at some case studies on that. But mutations, it's the main form of genetic variation. Genetic drift is a little different. These are chance events that cause a change in the allele frequency from one to the next. So it's most significant in smaller populations of animals. It can lead to a loss of genetic variation. It can be harmful, and those alleles can become fixed. This does not produce adaptations. It is just a chance event caused by a change in allele frequency from one that causes a change in allele frequency from one to the next. This could be something as simple as predation, or we open up hunting laws, right, in the United States, and now we can go out and hunt. So it's going to take it's a chance event that causes a change in allele frequency. So we're going to take out part of this population. There's two types. There's a bottleneck effect and a founder's effect. We'll go over each one. Pause here to take notes. Awesome. So bottleneck first. It's called a bottleneck, as you can see over here, because it takes a larger original population and then an event occurs and there's only a small proportion of the surviving population. So it's when large populations drastically reduced by a non-selective disaster. This is floods, famines, fires, hurricanes, hunting, all of that is a non-selective disaster. Floods don't care if you are, if your fur is a different color, they're gonna wipe you out either way. Some alleles may become overrepresented or underrepresented or totally absent, right? So you can see up here, there's that darker red, but you can see in the surviving population, there's no dark red. So, but there's a heck of a lot of this like pink color, right? So it's overrepresented. The darker red is underrepresented. Actually, dark red's absent. It's that green that is underrepresented. You only got two. Right, so you can see it can be devastating to a population. Founder effect. Yeah. Sorry, I'm covering study hall right now. So founder effect. Great. It is similar, but it's when a few individuals become isolated from a larger population. They establish a new population with a gene pool that differs from a larger population. We see this all the time on islands. So you will have a larger population, a parent population on the mainland. There's a migrating population that moves to an island biome, and the population after a couple generations changes and evolves over time. We call that the founder population. So it's the founder's effect. So we're removing individuals to become isolated. One of the greatest examples of this, I don't know if this would be considered founder's effect, is Lake Victoria over in Africa. It was originally one large lake, and then the water level went down and it produced three separate larger lakes in which those populations went on to evolve independently of one another. It's very interesting. We'll take a look. Okay, so founder's effect. They go and they found a new place to live. This loses genetic diversity because, as you can see, we start over here with the pink or with the red and the purple, and then eventually it's all purple. So it is a loss of genetic diversity. Great, let's talk gene flow. Gene flow is a transfer of alleles in or out of a population due to fertile individuals or gametes. So they could be transferred between populations. This happens a lot in migrating birds. So we see migrating birds down over here. We got the blue guy, we got the red guy. When they come together and they mate, right, you have offspring of the immigrated bird is now heterozygote. Offspring of the bluebird over here is now heterozygote from population A to population B. Now, when they mate with their own populations, that gene flow is going to show up, right? Pollen is a great example of this as well. It's essentially just moving gametes from one to the next, right? So we are migrating to a different part of the world and mating with like species over there. Do a quick review, examine each scenario, and determine if it's an example of bottleneck, founders, or genetic drift. Justify each response with an explanation. So let's do this together. Scenario 1. The black robin is a small bird native to the Chatham Islands in New Zealand. Before the 19th century, much of their habitat was converted to farmland. Due to the loss of their habitat and the introduction of non-native predators like cats, who is a non-native predator in the entire world, the population declined to its lowest point, only 5 individuals. Then, conservation efforts brought the population number back to an estimated 230 individuals. So, big points here: loss of habitat, introduction of non-native predator. What kind is this? Is this bottleneck, founders, or what was the other one? Genetic drift. Write it down. Bottleneck. Great. In 1720s, an Amish population settled into Pennsylvania. A number of the early settlers were from Germany and carried an unusual number of mutations, one of which included polydactyl. Interesting. The trait for polydactyl is now much more common in the Amish population than it is in the general population. Polydactyl is having 1, 2, 3, 4, 5, 6 fingers. Awesome. Is this genetic drift? Is this bottleneck or founders? Write it down. Founders effect. Tell me why. Scenario 3. Recurrent flash floods, so natural disaster, has caused mass mortalities in the marble trout population. Scientists have analyzed the genetic makeup of the remaining marble trout population and has found that genetic diversity has decreased significantly. Is this bottleneck, founders effect, or genetic drift? If you said bottleneck, you are right. Tell me why. Scenario 4. Due to an increase in wind in the local area, pollen from one population of flowers has traveled further than usual and pollinated another population of flowers. The offspring of these flowers now have traits of each population. Is that founders, bottleneck, or genetic drift? Gene flow, genetic drift, absolutely. Tell me why. Write it down. Okay, so now that we're familiar with natural selection, we're going to take a look at how it affects the population. Reproductive success is measured by relative fitness. So however many offspring you have, that's how we determine how healthy a species is within a population. The number of surviving offspring that an individual produces compared to the numbers left by others in the population. So it's total surviving offspring compared to how many are left in the population. The effects of natural selection can be measured by examining these changes in the means of phenotypes. So there are 3 modes of natural selection: directional selection, stabilizing selection, and disruptive selection. We're going to go over each one individually. I promise too, this is going to be a question on the AP exam. So let's take a look. Modes of natural selection. So you can see the red is before selection and the blue is after. Directional selection is a selection towards one extreme phenotype. So more of this trait, let's say this is beak length in finches. The 6-inch beak is more preferable to that species and thus will be passed on to future generations than, a 3-inch beak, but for whatever reason. Yeah. Next, we have stabilizing selection, which is mostly what we see when we're looking at natural selection in the real world. It's selection towards the mean and against extreme phenotypes. So somewhere in the middle. And then finally, disruptive selection is selection against the mean, and both phenotypic extremes have the highest relative fitness. So this is like, well, the birds with the 3-inch beaks found their plant that works for them, and the birds with the 6-inch beaks have found their plants that work for him, but everyone in the middle can't find the right food source. Pause here to take notes. Very nice. Welcome. So then we have sexual selection. It's a type of natural selection. This is humans, right? This is why we see like big showy traits for many species in the wild, or like mating dances, or like it's interesting. Like, have you ever seen a female peacock? They're very boring. Often males have these useless structures, so like the colorful male peacock feathers, and the only reason why they have it is because females choose that trait. So selective sexual selection and passing those genes on is another type of natural selection. This can actually produce traits that are harmful to survival. Colorful feathers in male peacocks make it easier to spot for predators, so it's sort of biological risk assessment, I guess. Okay, pause here to take notes. All right, let's get into some math. So Hardy-Weinberg equilibrium, again, I promise you this is going to be a question on the AP Bio exam. It's a model used to assess whether natural selection or other factors are causing evolution at a particular locus. So this is talking locus of a gene. So you got those right there. At a particular locus. Determines what the genetic makeup of the population would be if it were not evolving. And then this is compared to actual data. If there are no differences, then the population is not evolving. So it's essentially a mathematical model similar to the chi-square analysis that we're comparing our expected results to our actual observed results and making a conclusion based on that. If there are differences, then we say that the population may be evolving. Pause here to take notes. So the Hardy-Weinberg principle states that the frequency of alleles and genotypes in a population will remain constant from generation to generation, provided that only Mendelian segregation and combination of alleles are at work. So this is true only if we're talking about Mendelian segregation and recombination of alleles, so only dominant and recessive. This Hardy-Weinberg, unfortunately, cannot be applied to humans. We're too complex, so these are very simple species we're talking about. Remember, this is a hypothetical situation where no evolution would take place. In real populations, the allele and genotype frequencies do change over time. So this is like a model that we can use if we all get together and say, okay, we're going to hit pause on whatever this population's doing and take a look at it. There are 5 conditions that must be met in order to have Hardy-Weinberg equilibrium. So 1, there's no mutations. So there's no mutations going on within the population currently. There's random mating, so no sexual selection. There is no natural selection going on. Extremely large population size. This does not apply to small populations. There's no gene flow, so no members of the population are going in and out. If any of these conditions are not met, then microevolution occurs. That's the mutation, gene flow, genetic drift, natural selection, and non-random mating. Okay, so we are looking at evolution on a macro scale. Turn to someone in the room and discuss with a partner how each of the 5 Hardy-Weinberg conditions, if not met, can lead to evolution. So, turn to your dog or your cat or your parents or your friends or your auntie and explain to them how each of these 5 Hardy-Weinberg conditions, if not met, can lead to evolution. They're going to think you're crazy, but you should do it. All right, do that. So, math, 2 formulas. This one's an easy one. P plus Q equals 1. Here's the more complex one. P squared plus Q squared equals 1. The P is going to be the frequency of the dominant allele in the population. Remember back to when we were looking at gene pools. So big B, little b. Big B is P, little b is Q. It's the frequency of the recessive allele. So this makes sense that P squared is your homozygous dominant. Your 2PQ is the percentage of heterozygote, and the P squared is the percentage of homozygote recessive, right? Because if P is dominant, right, capital P, so that's P squared, that's homozygote dominant, okay? So P is dominant, Q is recessive. Great. So quick check. You are told that 20% of a population is homozygote recessive. What variable are they referring to in the Hardy-Weinberg equation? Homozygote recessive. Boom, Q squared, you got it. You are told that the frequency of a dominant allele in the population is 75%. What variable is that referring to in the Hardy-Weinberg equations? It refers to P only. We are not talking about homozygote recessive. We are talking about a single dominant allele. See the distinction? Love it. Okay, so here's how we work with it. Which formula you start with depends on the information that you're given. If a problem gives allele frequencies, then we're referring to P and Q. If it talks about an individual organism or the population as a whole, we're looking at P squared, 2PQ, and Q squared. Okay, so you see allele frequencies, you're doing the more simple equation. If you're looking at individual organisms or total populations, you're going to be using the more complex equation. A lot of times you're going to use both. Usually you're given Q and then you need to find P. But you're also going to see problems that that's not the case. What's that? On the desk over there. Take down the chair that is on the desk with the tissues. All right, some tips for solving these problems. Always write down both equations. Identify the information given. Is it for alleles or is it for a population? If it's alleles, then you're using the P plus Q equals 1. For populations, you're going to be using the P squared, 2PQ, Q squared. You're going to be using that equation. A lot of times you'll use both. Write them both down. Regardless of what the problem asks for, solve for P and Q first, because that will allow you to answer any question. If you have P and if you have Q, the rest of it is plug and play. Use your calculator to do the square and square root functions, right? Don't make simple mistakes. Don't assume you know the answer. Use a calculator for everything. That's why we give it to you. And double check your work. Okay, we're not going to watch the video by Mr. Anderson. If you would like to watch it, here it is. I will also post it on Classrooms. You can type into Google Mr. Anderson, Hardy-Weinberg problems, but the sound doesn't really come out, so we're going to skip it. Practice problems. Yeah, you're going to do it. So you have sampled the population and found that 36% are homozygote recessive. What is the frequency of the dominant and recessive alleles? So, excuse me, following the steps, we're going to write down our two equations. And we are given that Q squared equals 0.36. That is from the 36% are homozygote recessive. So they're given that to us. Remember to solve for P and Q first regardless. So how are you going to solve this? Well, first you're going to take the square root of 0.36 because that will give you Q. Then you plug it in to P plus Q equals 1 and solve. So P is 0.4 and Q is 0.6. So the frequency of the allele, of the dominant allele, because remember this one, they're only asking for dominant and recessive. They don't care about the heterozygote. okay? Allele A is 40%, P is dominant, and the frequency of the recessive is 60%. Remember to put your units. It's not 0.4 anymore. You're going to report it as a percentage. Cool. Next up, 10% of a population of mice are albino. Albinism is recessive to tan coloring. What is the frequency of homozygote dominant and heterozygote individuals in the population? Step 1, write down your equations. Step 2, figure out what's given. So albinism is recessive and 10% of the population has it. So that's our Q squared. We put it into a decimal form and we solve for 2PQ to get our heterozygote individuals, our homozygote dominant individuals. So here's what we do. We're going to, remember, step 1, regardless of what you're trying to figure out, work out your P's and Q's. So P plus Q equals 1. We're going to square root our given value, and we'll solve. So with Q being 0.32 or 32%, and P being 0.68 or 68%, now we could just plug and play, right? So we plug this in for square values. P squared is 0.46, and then 2PQ. So you're just plugging in here. Your Q goes in here, and your P goes in here, 0.44%. And then you're going to want to check your work. Right, so P squared is 0.46 plus 0.44 plus 0.1. You add those up, they all equal 1. So the frequency of homozygote dominant is 46%. That's what we get up here, homozygote dominant. P is dominant, squared means we have 2 of those alleles. The frequency of heterozygote individuals is 44%. That's the 2PQ, right? Because you have 1 of one individual and you have, or one allele and one of the other allele, 44% and 46%. We didn't care about the percentage of Q of that recessive, but we needed it to find the other 2. Awesome. Practice problem 3. A recessive genetic disorder occurs in 7% of a population of birds. What percentage of the population is homozygote for the normal condition? Step 1, write down your equations. Step 2, write down what you're given. 7% is 0.07, and we know that is recessive, so that's going to be our Q squared. And we need to solve for P squared, right? Because they are asking homozygote under normal conditions. So when they say under normal conditions, that means that they do not have this rare genetic disorder. So normal conditions would be homozygote for normal conditions. So homozygote for dominant. So, but we have Q, so we're going to work it out. So we got the square root of 0.07, 0.26. P plus Q equals 1. So we can find P here. We just plug and play. And then you're going to want to square it. That's it. So we got 55% of the population is homozygote dominant. Love it. Practice problem 4. Population has 1,000 individuals. 132 of them are homozygote dominant. 455 are heterozygote. 422 are homozygote recessive. What are the frequencies of the dominant and the recessive allele? Step 1, write down your equations. So the given is uncalculated, but they gave us everything we need, right? So out of 1,000 individuals, 123 of them are homozygote dominant. That would be our P squared, 455. So we just need to calculate those. And then solve for P and Q. So the first thing we're going to do is get the homozygote dominant into a percentage, right? So P squared, so 123 over 1,000 equals 12.3%, right? Or 0.123. Then we find our Q squared. Then we find our 2PQ. Okay, so, but that's not really what we're looking for, right? We're looking for the frequencies of P and Q. So now that we have P squared and we have Q squared, we can then square root it and find the answer. So the frequency of alleles here, the dominant allele will be 35%, and the frequency of the recessive allele is 65%. And if that sounds confusing, that the recessive allele is going to have more, remember, we are doing the alleles individually. We have that right there for the heterozygote individuals. Cool. So, quick review. What's the difference between genotype frequencies and allele frequencies in a population? If you said that genotype frequencies refer to how alleles combined and allele frequencies refer to how alleles' relative distribution in the population is, or how common the allele is, you're right. Hardy-Weinberg equilibrium is often referred to as a null model of evolution. What does this mean? The null model of evolution is one where no evolution occurs, right? Remember those 5 conditions met with Hardy-Weinberg? One of them is that it's not evolving. No random mutations, random mating, all that kind of stuff. Great. So, variations within populations. The more genetic diversity there is in a population, the better it can respond to environmental changes. Remember, we're talking about whole populations, not individuals. More likely to be individuals that can withstand changes, right? So if you have a whole population here and there's a wide range of variation within that population and a wide range of genetic diversity within that population, and some natural disaster were to come along, more of that population will survive just purely based on the fact that there's more variation in it. Species with low genetic diversity are at risk of decline or, at worst, extinction. So, for example, this is actually interesting because the California condor has made a resurgence in the past few years. Due to poaching and poisoning, the California condor was reduced to 27 individuals. This drastically lowered the gene pool. Even though restoration efforts have increased the numbers, they have lost their diversity. I think California condors now are up in numbers. But yes, 27 individuals is too small for a population. That's it. So, a little bit of a longer topic, but we're done with it. Great. I will see you in class tomorrow.